Monocot embryo development. In angiosperms, mainly two types of embryos are here. One is dicot embryo, second one is monocot embryo. These two embryos are belongs to angiosperms. Monocot embryo development discussed today. See, monocot embryo development identified in Sagittaria species. Sagittaria species belongs to monocots. That monocot embryo development identified in Sagittaria species. See, the Sagittaria species belongs to grass family. See, we know fertilization. True fertilization after diploid zygote is formed at the micropylar region of the embryo sac or ovule. True fertilization means one male gamete fuse with egg cell to form diploid zygote. The diploid zygote is also called oospore. It first identified by Strasburger scientist in angiosperms. And the true fertilization is also called syngamy. After syngamy, one diploid zygote is formed in the ovule. The diploid zygote first participated in the transfer division to form two cells. Here it is, CA, CB cells are present. CA cell present at the micropylar region, CB cell present at the chalazal region. Only two cells are present initially. From the two cells, CA cell develop into suspensor. It enlarged into large size, expansion of size. It act as a hustorium. Hustorium main function is collecting of nutrients, food material from the endosperm and pass to the developed embryo. See, CA cell cannot participate in the further division. It act as a swollen structure cell is called suspensor. It modified into hustorium. But CB cell also there. The CB cell again participated in the transfer division to form middle cell and terminal cell. Middle cell is also called D cell and the terminal cell is also called C cell. See, terminal cell first division participated in the transfer division, again participated in the longitudinal division to form four cells. See children, these four cells are developed from terminal cell, C cell only. The four cells again participated in the mitotic division to form 8 cells, 16 cells. That 16 cells again participated in the mitotic division. Before that participation of 16 cells, the 16 cells are arranged in a two orals. Outer oral 8 cells, inner oral 8 cells. Outer oral 8 cells take rest few minutes of time and inner oral 8 cells again participated in the mitotic division to form 16 cells. Total how many cells here it is? 24 cells are present here. From these 24 cells, outer oral 8 cells act as a dermatogen initial cells and middle oral 8 cells act as a plerome initial cells, peribellum at center region. And these are the three orals. Outer oral 8 cells develop into epidermis of shoot. Middle oral 8 cells develop into cortex of shoot. Inner oral 8 cells develop into vascular portion of shoot. See, this total portion is developed from which cell here it is? C. C cell participated in the transfer division, longitudinal division to form 4 cells, 8 cells, 16 cells. That 16 cells are arranged in the two orals. Outer oral 8 cells act as a dermatogen initial cells. They take rest time for the divisions. And below these dermatogen initial 8 cells are present. That 8 cells again participated in the mitotic division to form 16 cells. Total 24 cells are present in this stage. Outer oral 8 cells act as a dermatogen initial cells, and middle 8 oral cells develop into cortex initial cells, and center oral 8 cells develop into vascular initial cells. See, cortex, epidermis, vascular region belongs to shoot only. The complete shoot part developed from which cell here it is? C cell. The C cell is called terminal cell. At the same time, the C cell or terminal cell divisions, the parallel divisions also occurs in the middle cell. The middle cell is also called D cell. See, D cell participated in the transfer divisions only to form two cells. The two cells again participated in the mitotic division to form 8 cells. The 8 cells are arranged in the two orals. Upper oral 4 cells are present. Inner oral 4 cells are present. Upper oral 4 cells are developed into root cap. And below these root cap, root apex cells are present. See, these parts are developed from the D cell. These parts are developed from the D cell. Finally, C cells and D cells are rapidly participated in the divisions to form a globular-like structure of embryo. See, globular-like structure of embryo containing only single cotyledon. The single cotyledon is also called scutellum at the matured stage. So, monocot containing how many cotyledons? Only single. Single cotyledon is present. On the lateral side of single cotyledon, palmulae is present. Palmulae means shoot apex. Palmulae means shoot apex is present. And below these cotyledon, radicle is present. Radicle means root. Root part is called radicle. Finally, matured embryo containing only two major parts. One is epicotyledon. Epicotyledon contains cotyledon and palmulae. And the lower part is called hypocotyledon. Hypocotyledon containing only radical part. At matured stage of monocot embryo, suspensor is looses from the embryo. Finally, the matured embryo containing how many major parts here it is? Two major parts. One is epicotyledon and second one is hypocotyledon. Epicotyledon is the upper portion of monocot embryo, and hypocotyledon is the lower part of the monocot embryo. Now, in epicotyledon, shoot apex is present. That shoot apex is protected by one protective layer. Shoot apex is protected by one protective layer is called coleoptile. Lateral side of the shoot apex or a lateral side of the coleoptile, one single cotyledon is present. That single cotyledon is also called scutellum. See, reduced cotyledon is called epiblast. Reduced cotyledon called is epiblast. These parts belongs to epicotyledon. Epicotyledon means upper portion of monocot embryo. At the same time, lower portion of monocot embryo is called hypocotyledon. Lower part is called hypocotyledon. Hypocotyledon containing root called radicle. Radicle containing root cap. The root cap and radicle is protected by the one protective layer is called coleorhiza. Coleorhiza is the protective layer of root. Coleoptile is the protective layer of shoot. So it is the matured embryo of monocot.